Welcome. This is so fun to kick off our new year with all of you filling this room. And you are going to be so glad that you came today because we've got a special treat with our speaker. The Center for Brain Health. My name is Dr. Sandy Chapman. I'm the founder and chief director. I'm a professor in the School of Behavioral and Brain Science. I have dedicated my life to helping individuals across the lifespan find ways to make their brain stronger, longer. For those of you that are online, we have an online audience. There will be time for question and answers. Please use the Q&A code to fill your questions in. I'm sure there's going to be lots. I hope you give us time for questions, Dr. Burton, because you're going to create, I'm not sure he did, but we'll see. As well as the in-person, raise your hand with the questions. For those of you that don't know about the Center for Brain Health, we are dedicated to the neuroscience of defining what is brain health, measuring it, helping people find ways to enhance it, and to find ways to scale that work. So we don't want to just be a silo here. We want the whole world to do that. We're already in every single state in 65 countries, and we hope that you're taking your brain seriously. But what I'm most excited about today is that finally, I've wanted Dr. Michael Burton. He was featured on UTD's website. I've known him since he started here, but we're very lucky. He's a Eugene McDermott Distinguished Professor. He is a founding member of the Center for Advanced Pain Studies in the School of Behavioral and Brain Science. He has already been selected as a fellow of the Mayday Pain Society. Every one of us have pain at different times. He's one of the pioneers to help us understand parts. He's also interested in communicating science and improving care. Dr. Burton's research focuses on the interactions of our immune system and our nervous system. We know that these systems talk to each other. Well, too often they are separated. Thank you for putting those together with your innovative work. And he looks at regulating pain and energy homeostasis. He studied the peripheral stimuli, painful stimuli, diet, alcohol, and immune activation. Dr. Burton's lab studies aging, nutrition, alcohol consumption, and immune activation. His bachelor's and PhD were in animal studies at the University of Illinois in Urbana-Champaign with a focus on immunophysiology and behavioral. He came to Dallas as a postdoctoral fellow back and worked in the Department of Hypothalamic Research at UT Southwestern. While he was there, he continuously gained skills in molecular genetics, neuroendocrinology, and neuroanatomy. What don't you know? Mercy. He then, fortunately, we were able to grab the talent and hire him at the University of Texas at Dallas. We're so fortunate. He focuses on the way immune cells influence the transition to chronic pain, bioinformatics, and in vivo two-photon imaging. He received the distinguished NIHK award. What I especially appreciate about him is he wants to make sure his science is translated to help humankind. Please join me in welcoming Dr. Michael Burton.
All right, thank you so much for that wonderful introduction and for the invitation to come and share with you all a little bit about what we do up in Richardson. All right, so I'm Dr. Michael Burton, and I'm going to talk today a little bit about something that I go back and forth with about how important it is and then how impactful it's going to be. It's about alcohol consumption and how alcohol consumption actually changes our immune system and behavior because of it. But before that, I will just give you a little outline. So I always like to talk a little bit about myself because whenever I come to venues like this, I just don't know how much people know. And I'll talk a little bit about the lab and then go into like our approaches to things that we do, pain, and then alcohol-induced pain states, and then cell interactions to a little bit at the end because I have to be as kind of baseline as possible. So some of my training expertise, like Dr. Chapman was saying, is immunophysiology and behavior, where I went to the University of Illinois. And then I came down here and got my senses and got to Dallas as soon as possible. I always tell students, I'm from Chicago, but I got here as soon as I could. And what we do in the lab now is try to understand everything that I learned across my whole life and put it all in one box, which is really difficult to do in some cases. But I always like to talk a little bit about kind of how I got there. And what happened is, growing up in Chicago, I was single parent household. My mom did the best she could to raise myself, my brother and sister. And most people think that they're going to get out of these types of situations by doing things like playing athletes or doing athletics. And I thought the same thing. I was decent enough to be able to walk onto the team at the University of Illinois, and then decent enough to get hurt within like a month after I got there, which is really a bummer because I walked onto the team. And I earned a scholarship, so I got a scholarship for a semester, and it disappeared as fast as it came. But then it left me with this kind of chronic inflammatory injury, and that injury had me kind of thinking, why is this the case? It took about 6 months for it to heal, and then when it finally healed, I still had pain. It was like, well, everything's resolved, but I'm still dealing with this all. This is something that is like a chronic state with a lot of people where after the resolution of a particular injury or inflammation, you still have pain. And so that kind of sent me down this journey of like trying to figure out why that happened. And I talked to a couple people as an undergraduate, and one of them was Rod Johnson. He said, I really care about how the immune system impacts your behavior and inflammation, and so we should do it together. And so I actually worked with him for like as an undergraduate and then as a PhD, and we still work together to this day. He's such an important person to me. Got here to Dallas and worked with Joel Almquist. I'm not sure who knows who that is, but I told him that I wanted to do pain, and he said, I don't really do pain. I'm a vet by training, and I'm not going to do anything like that. You have to find someone else who's decent in pain. And so I ended up finding this guy named Ted Price, you may or may not have heard of him, to work as a postdoc with him for a couple of years, and he kind of taught me how to integrate all the things I knew about the immune system, genetics, and then pain and metabolism. So the lab now focuses on these integrative approaches. I would call us a translational lab with a lot of preclinical, so there's some preclinical data here. But then we also have some clinical data as well, looking at how lifestyle factors like diet, age, and alcohol really influence this communication and why it's important. We opened the lab, the doors in 2017. We have undergrads. I did my tenure packet a couple years ago. I had 120 undergraduates in the lab in 5 years. Grad students, PhD students, postdocs, all types of people. It's been an absolute blast. I think they're watching online. I'd like to talk a little bit about my lab philosophy before I really get going, because I think it gives people more perspective of me, and then I'll start talking about science. But there's kind of two big parts. The first part is kind of a slogan from Google's early days called "Don't Be Evil." And the idea is that everybody should have all the information that they possibly could ever need, but just don't use it for evil. And so for us, I say everybody in the lab has access to all the information and resources that we can, so that we can make complete and educated decisions about what we do within our space. Then the second thing is to be deliberate. And indifference to evil is worse than evil itself. In a free society, some are guilty, but all are responsible. And this was a saying by a guy by the name of Abraham Heschel, who was a rabbi, theologian, philosopher, and a civil rights activist. And I still think, and this was said in like the 50s, so it still holds true in whatever space. Good science is for everyone and should be shared absolutely, absolutely. And so that's a little bit about me. Like I said, I like to try to do that, a little primer, before we start getting into some more nitty-gritty. And so what my lab's focus and our focus is trying to understand this interaction between the nervous system or neuroscience, psychology or behavior, and the immune system. Most people have heard of all those things, but not necessarily to like a complete degree. And I always tell students and trainees that join my lab is that you're in for a wild ride, because this is not just one thing. We're learning essentially 3 different disciplines. And so what it is what happens in the immune system and how that impacts your nervous system. And importantly, what happens is your immune system, your nervous system acts like an immunostat, like a thermostat. You want a set point to be healthy and normal. However, things happen, you get sick, you have to change your behavior because of that. It's really important that that happens, because if you don't change your behavior, you can get more sick and things can happen in a negative way. However, and like the case of my injury, when I got hurt and the pain didn't necessarily resolve, but the inflammation did, there's dysregulation in that, and we're trying to understand really kind of what the demystifying what's going on there. So the focus of the lab is really, again, understanding neuroimmune interactions, but in the context of all these factors that are really important that you may not necessarily always get to measure. So for instance, social factors, psychological factors, different comorbidities. If I'm depressed or slightly burned out, the video before is talking about burnout. So I'm not necessarily clinically depressed, but I'm just real stressed. How does that change how I behave? How does my immune system influence that, right? And so really we want to understand how all these mechanisms come into play. So we have three major projects in the lab. I'll talk about one of them today. One is like in mechanisms of aging and evolution. And the other one is the circuitry, like what cells are doing what and where and why. And then lastly, we were actually interested in the role of something called cannabinoids, which I'm sure people have heard about, in mediating inflammation and pain behavior as well. And in all cases, we use mostly animal models, but we also have some human donor samples, patient-derived data, and clinical data as well. So what's our approach? Again, we really want to think about pain, because pain plasticity pathways are very important. So you can have an adaptive pathway, meaning I touch something that's hot, I remove my hand because it's hot. You need to do that, right? Your body needs to avoid that damage that can come from it. However, in my case again, when I'm dealing with chronic inflammation or chronic pain because of the inflammation that's been resolved, it's a really negative situation. And so we really want to understand how you can go from adaptive, where you need it, to maladaptive, where it's problematic. And so this whole highlight here of acute to transitional pain is really the hyper focus of the lab, right? Because you go from one point to the other. And in doing that, we want to identify identifiable targets and cells particularly that are non-normal in nature. Currently, what we're using now, they work short-term, opioids. They really do work short-term, but long-term they don't. And really the problem that we find is long-term pain is the most problematic. So then in order to do that, we have to understand this neuroimmune conversation, or unique back and forth dialogue between cells of the immune systems and cell of the immune system and cell of the nervous system. And then lastly, something that I'll touch in a second, is to understand how cell types can mediate sexual dimorphic action. So how males and females can deal with this differently. Why this is important is because about 50 million Americans struggle with chronic pain regularly, and about of those 50 million, 70% of those are women. There is this really lopsided measure of how women are experiencing pain states. Nobody really knows why, and so that's part of what we were trying to do and figure out. While opioids, again, are effective in the short term, roughly 25% of patients prescribed opioids for chronic pain misuse them, overuse them, become tolerant to them. There's many, many ways that it happens, and potentially overdose on them as well. And since the COVID epidemic started, there's actually been an increase in opioid-induced deaths, where back in 2021, there's about 121,000 people that died just from opioid overdoses. And so can we discover mechanisms that are going to be important to employ efficacious non-opioid-based therapeutics for the treatment of chronic pain is our goal. All right, so I'm going to pivot a little bit and talk about lifestyle factors. And so, and an important lifestyle factor are the things that we put in our bodies. And one in particular is alcohol. So food substances like high fats, alcohol, have been shown to play a role in chronic pain in some way since the 80s. However, over the past 30 years, there's been a 27% increase in the amount of actual alcohol consumption in pretty much every country, and more people worldwide suffer from pain, peripheral health disparities, alcohol use disorder, and comorbidities like depression more now than ever. And of this, about 100 million people worldwide have some type of alcohol use disorder, and 40% of them actually experience pain. And I'll say it again, because it's a huge number, right? 100 million people worldwide have some type of alcohol use disorder, and 40% of those people develop chronic pain. So this really points to this really interesting bi-directional communication between, and multifactorial communication between many places that are really important, and we need to really pay attention to it. So everybody says everything in moderation, right? We've heard that before. Everything in moderation, alcohol in moderation is good. Talk about the battle. I sit in front of people and always say, do I really want to tell people alcohol is bad when we use it for like social lubricants and things that we use it for? And so I was reading this New York Times article a couple years ago, and it was talking about just that. Many other countries already talk about or tell their citizens alcohol is bad. In fact, even a little bit of alcohol can harm your health. And this was an article written in 2023. And importantly, after decades of confusing and sometimes contradictory research, too much alcohol is bad for you, a little bit is good, and actually even small amounts of alcohol can have pretty severe health consequences. And then just this year, a few weeks ago, the Surgeon General called to slap cancer warning on all alcohol, just like cigarettes. So did we know? But do we know, right? And so it really comes into play. The more you know, the better. Everything in moderation is important. But what I want to talk about just a little bit today is some of the early findings that we found that even a little bit of alcohol can be deleterious. Okay, timing is very important. When I say timing, I mean thinking about when you are developing a drug and you want to help people, or you're trying to diagnose people. I like to think about Parkinson's. A lot of really interested in Parkinson's. We're trying to get more into it because, come to find out, many pathological conditions have an overpathology at the very end where diagnosis is really straightforward, but treatment is almost impossible, right? Because you're just so far down that road. However, thinking about what we call prodromal or early phases, like overlapping symptoms and mechanisms, maybe we can assess the development of these pathologies like Parkinson's and think about ways to actually target them therapeutically. And I'd like to use this as an example of there's two stages, pre-diagnosis stage and then Parkinson's disease stage. When you have motor symptoms and treatment complications when you're really far over here. However, there's a number of these pre-diagnoses factors, which I'm not sure you agree, but I'll tell you. Sensory systems like unspecified pain, other symptoms like impaired olfaction, depression, and cognitive impairments are all within this pre-diagnoses phase. And so is there a way to think about this and pre-treat or think about treatments and mechanisms to help people? So that's what we're thinking. So with us, the idea is mechanisms of chronic pain development. Can we look earlier before the onset of chronic pain actually happens to determine mechanisms behind its development? A lot of research is kind of focused on this side of the kind of pyramid here, and we're trying to be here. And so there's not a ton out there, but we're going to try to be the ones to help make the literature and make more of it. So pain development and alcohol habits. Now, we know that, or we've been told that a drink a day, a glass of wine a day, keeps the doctor away like an apple will do. And so the question is, does short-term moderate alcohol consumption alter pain sensitivity? And the impetus for this was, we had a study where we were looking at high fats, and we saw that if you ate a really high fat diet, you necessarily didn't have pain, but what happened is your pain sensitivity level changed. And it makes sense because there's low-grade inflammation that's happening when you're eating this high-fat diet. And so we were like, was the same thing happening when you think about alcohol consumption? And so the thoughts were, what's on the screen here is that if you are drinking alcohol and across time, there's an event called priming, which essentially your body's in a normal state, but then you're drinking and it becomes not so normal and more inflammation, and as time progresses, you reach to a point where your body's now altered. Meaning, if you experience something that is normally not painful, a stab or some type of inflammatory thing at the tip of your finger, but it goes away typically. But if your body's already primed because you've been having that diet or drinking too much alcohol, that actually causes really, really robust pain and changes exactly how you feel. And so that was our kind of thought and goal on this all. And there's words on here that matter and that don't, but essentially we use something called an inflammatory agent called prostaglandins, which you may have heard of in the context of headaches. And this word, allodynia, what it essentially means is it changes how your body senses an actual sensation. So it's usually taking a sensation that's not painful, but now you feel that it is painful. So allodynia is changing from, I don't feel like this is pain at all, to all of a sudden, for some reason, this is painful. And so we want to understand these mechanisms in that. And so the objective, we'll go into a little bit of data, is how to look at short-term moderate alcohol use, and does it cause a change in sensitivity? The first thing was, does it cause pain at all? And I think if it doesn't cause pain, that usually lets people know that there's not a problem. The number one reason why people seek medical attention is because something's painful. Something's not right, right? And so that was the first thing. And then the other thing was, are there changes in allodynia? So something that's not painful, now is it more painful? And that one is harder to tell, right? Because there's so many dynamic factors that are happening in your life that you may not be paying attention to the fact, oh man, it does hurt a little bit more than it should. But I'm gonna keep it pushing because it's not that bad. So there's particular machinery in your body that's involved in this, and it's called a hyperalgesic priming machinery. And what it essentially does is if you have a normal scan or a normal paw, and you inject with this really low threshold of a prostaglandin, you feel it and it goes away pretty quick. However, if you're primed, meaning that your body has dealt with something and now your sensitivity's changed, you have an increased or an exaggerated pain response, but it's also protracted and lasts longer. And so how are we going to figure that out? And without going into dramatic details, we figured out, or people figured out, that there's cells in your spinal cord that are really responsible for mediating that shift from acute to chronic pain, mediating that hyperalgesic priming. They're called PKC neurons. And so it's complicated to do this type of experiment with people. We're going to try, but now we're doing it with mice. And what we're doing is we're saying we're going to put a mouse and let them drink alcohol for a couple weeks. Nothing crazy. We call it the spring break model, right? And so super low grade. They get similar calories from everything else, except for they just have a little bit of alcohol. And it's a really widely studied model. People have been doing it since the 80s. And the animals really like it. So go figure. My graduate student who actually just recently graduated a couple weeks ago, we're trying to get her dissertation completely done, congrats, Siobhan. She worked on this for several years, and she worked with mice and trying to understand how their pain behaviors changed and how comorbidities like depressions changed. And what she would do is she would acclimatize or acclimate the mice to this diet, and then put them on the diet for 14 days. And then she would assess pain measures and other behavior measures and take tissue like the spinal cord to see if those cells in the spinal cord actually changed. And so an early study where she was looking at the effect of mechanical allodynia after 14 days, what she saw is that in males, there was a decent effect in this blue line, but in females, there was a pretty robust change in the actual effect size of PGE2-induced inflammation and pain. And when she looked a little deeper, and she looked in the spinal cord next to those PKC neurons, she was looking at immune cells that were active. And when she looked, she saw that when she looked on the side where the pain was supposed to be, she saw way more active immune cells hanging around those neurons that were really, really inflamed. So the neurons themselves were there. They weren't doing that much, but the immune cells that were there were really active and really responsive. So to us, that's telling us that the immune system is completely affecting what's going on in the spinal cord and the nervous system by leading to this pain state. And so she and I sat down and we were we're talking about, like, what about these specific immune cells is important? And so we're gonna get a little bit more granular. We're gonna try not to get too bad. And so what we found in the literature is that people that are drinking alcohol and alcohol abusers, there's different parts to the alcohol. There's the alcohol itself. It gets broken down to different components, acetaldehyde, et cetera, et cetera. I can talk about chemistry. But it turns out that all these components react and interact with immune cells at this particular receptor called toll-like receptor 4. And if it's early alcohol, if it's been drinking alcohol, if it's chronic alcohol consumption, and this is what all these models are, what they found is that these immune cells, which are called microglia, were reactive to the alcohol itself. So you drink alcohol, people talk about a blood-brain barrier, can alcohol pass it? Absolutely. Passes it freely, it interacts with those immune cells and so forth. So we wanted to figure out a way to really prove that that's actually the case. Are these immune cells really responsible? And so we hypothesized alcohol is metabolized to activate this microglia TLR4, and this in fact drives the initiation of alcohol-induced allodynia in females. There's so many questions that we have, and I'm curious to see what everybody else's questions are. So the problem with this marker is that it's expressed in a lot of different cells, and so there's really no way to test it out in a human, so we have to use a mouse again. But this time what we can do is generate a molecularly different mouse that lacks that receptor anywhere else except for the cells that we care about. And why that's important is because it lets us really, really nail down, are these cells and that particular receptor on those cells vital? If they are, then we can use that as a therapeutic tool. So, I won't bear pause you guys with the details of all that. But in literature, we usually go and ask questions, is this gene required or is this gene sufficient? And what that requirement versus sufficiency thing means is, if I have this gene, and this gene is on the cell, is it very, very important to the actual outcome? Or is it just part of the outcome? Versus sufficiency is it all about the outcome? Is it because of this gene on this cell why we have this problem? And so we chose to use that route as opposed to asking if it's part of it or not, because we really want to know if we hit this thing with a hammer, is it really going to have an effect? And through reading and background literature, we knew that we had a strong idea that was going to work. But in science, you just can't tell people you're going to do that and it actually be done. You have to prove all the things that you're doing and why. And so these are just a couple of things that we did just to prove that these cells that were in a wild type or normal animal had that gene. And then when we knocked out that gene, it was gone. And then when we put it back in that gene, it was back, right? And we looked at other places, like other cells, to make sure that it was gone from those cells. And it was, right? So doing all this, we wanted to just say, we absolutely have the cell with the gene that we need. And then we did the same paradigm again, where we gave the animals this alcohol for 2 weeks. Had them do a couple of assays, and I'll share the assays in a second, and then came to the output. So this is a typical graph that we show in the lab, and what it's showing is something called mechanical allodynia and Paul withdrawal threshold. You can't ask a mouse, hey, are you in pain? What's your pain 1 through 10? But we can assess different measures of how they respond. And one measure is if I slightly poke you with a toothbrush bristle, something like that, how do you respond? And typically it only takes about 1 gram worth of pressure for the animals to respond. However, if they're in some kind of pain, it'll take less than that. And so the further you go down on this thing, on this axis, the more pain you are in. So what we found is that in the females, that after their alcohol and their PGE2 injection, if they had that gene on that cell, they were significantly in more pain. And so when you remove that gene everywhere else, they didn't have that response. When you put that gene in that cell only, they had that response. It was pretty robust. And then when we compared it to just completely normal animals, we saw the same thing. And then strikingly, when we did the same experiment with the males, their brother counterparts, we didn't see any difference whatsoever between the males and the females. I'm sorry, between the knockouts and the animals with the gene in them. However, when you gave the males the ethanol, they did have some kind of response, but it wasn't near as robust as the females. So there's clearly this female-specific response that's mediating this kind of painful state. All right, so then let's look back at those cells, those immune cells, and ask the question, are they active? And so we look back in the dorsal horn within the spinal cord, and then we saw over here on the right side, females that were just normal wild type, and females that just had the gene in that particular cell, they were just as responsive. And to us, that meant that gene was sufficient and absolutely necessary for the responses that we're seeing. We didn't see that same level in the males. It's dynamic, and it's a little bit of something happening, but nowhere near as robust and clear. And then people always ask other questions like, do the mice get drunk, and do they lose weight, or what happens with everything else? So I was like, you know what, man, we'll just put it up here. So we checked for all that stuff. No, they don't get drunk. They can be drunk, but not in the model that we're using. They don't lose weight or change their weight at all. They're consuming roughly the same amount. Both the males and females are drinking the same amount. And they have the same amount of blood alcohol content level in the blood, right? So like it's not the men are drinking less or the males are drinking less or the females are drinking more, they're drinking the same amount and essentially have the same amount of alcohol. Then another question that we got a really good question from someone was, so alcohol can cause the gut to get leaky. And so could the leakiness be different in the males and females? We checked, no. And then what about liver damage? Is there something about how the alcohol is being processed? Is that changing pain outcomes? No. So we checked all kinds of things, and it seems as if there's no difference between males and females in how much they drink, how they respond to the alcohol, any other type of peripheral gut stuff, or liver stuff. So the summary really is that there is a female-specific allodynia or pain that is microglia specific or immune cell specific, and those active microglia are in fact at close proximity to the cells that are important for that acute to chronic transition of pain. But I always like to share more based on the time. I think I have the time to do it, right? Awesome. So we always talk about in the lab, one thing or another thing, pain only, but I mentioned something about comorbidities. And a huge comorbidity to alcohol consumption is depression. So we asked the question further, are these animals more depressed? Now I can't sit there and with a little ND like a psychiatrist ask these animals, are you more depressed? We have to do another measure to assess depressive-like behavior. And so, same paradigm, 5% alcohol across 14 days, so spring break, loving it. But then we do something called affective behavior measures. We did several, but one in particular is called a forced swim test, which means you put the animal in a bucket that has water. Mice can swim like crazy. It's not like they can't swim, but they don't like it, and so they try to get out of it. They're extra motivated to get out of it. And we keep it warm and they're fine, but they just don't want to be in it. To me, and to others, that is what we call a measure of anhedonia or motivation. And if an animal is more depressed, they feel more helpless or more changes to their hedonic activity. And so that means that they move less and they give up all this woe in the world. And so when we did this, we were looking for higher times, and this is the males on the left and the females on the right. And what we found is there's a lot of different dots, a lot of different things, but a couple measures is that in the females, if they were completely wild type or had that gene in those particular cells, they had an increased amount of depressive-like activity. And when you compare it to the males, they just didn't. And so us, not only are the females exhibiting this change in pain sensitivity, but they're also exhibiting early stages of depressive-like behaviors. And then I won't go into dramatic detail on all this, but we looked into the brain and different parts of the brain, and we found that if you look at the circuitry of prefrontal cortex and the amygdala, if you're familiar with the brain, looking for those same cells and inflammation, we found all those cells in all those brain regions in females were way more inflamed in various regions that were really important for depression. So some concluding thoughts are essentially that the alcohol is really, really important in understanding how our immune system and our nervous system interact. And it's even more important to consider how small amounts and small moderations can really lend to complete changes in what we're doing with our body. There it goes. So there's a dichotomy in neuroimmune activation and crosstalk, meaning there's a back and forth between neurons and non-neurons like immune cells. And particularly this gene on these immune cells drive both pain behavior and depressive-like behaviors, I would say in a female-specific fashion. And TLR4 and other cells are undoubtedly important, but I really need more money and time today to figure out how to do that. Really fitting with this week, but it's true. All right, so then this is a picture of all the people that make things happen, the family or the lab throughout the years. This is us here in the middle, Siobhan, the student that was working through all of this stuff. She just defended on the 6th, and that's us that day that she's defended. And then the cursory kind of, these are the people in the lab, these are people that we work with in other labs and other spaces, and support funds from the NIH, that's hopefully still real. The Rita Allen Foundation, ADVED, Mayday Fund's been fantastic with helping me with like my science communication. And thanks for your attention.
Michael, that was brilliant. Wow, I have so many questions, but I am going to open it up. First of all, we're having a Women's Brain Health Conference, and I'm like, we need you to speak, but we're full. But I'm like, oh my goodness, this is so vital, this information. Thank you for that work. So I'm going to let other people ask.
Hello? Hello, okay. Thank you for your presentation. So if one decided to stop drinking, how long would it take for the body to come back to normal as far as pain sensitivity?
Love that question. That's actually one of the things that we're looking into now and thinking about now is if this does change, there is a maladaptive process, is that process reversible? Or is there a point to where it's just no longer reversible whatsoever? That's what we need the money for. So I think I've had to guess there is a point, but I don't quite know what it is, but I have ideas and plans of what that looks like. So.
You almost wonder, do men need to not stop and women do? just thinking.
Yeah. Yeah, but that's a great question. I love that question.
I know of two examples of women. Alcohol was not involved for the women, but their husbands were screamers. Another one was an alcoholic, and both of them contracted cancer. Then there was the trauma of divorce and remarried. Second husband was a screamer. It wasn't necessarily screaming at her, just screaming about things. And what she said was her startle response was very high. Her immune system crashed. And I noticed that, and she had breast cancer. I noticed you had tamoxifen on one of your studies. Was estrogen any kind of a factor? And without estrogen, without alcohol for the woman, have you seen any examples like that?
Yeah, you just unloaded. That was a lot. Yeah. But yeah. So to answer the question, the last question is the estrogen. We assessed estrogen levels throughout all of our studies, and we did a correlation to try to figure out if the animals that were in more pain had different levels of estrogen. We found zero correlation, and it seems to be the case across a lot of studies where estrogen and the cyclicity of estrogen across the phase or not doesn't have as much of an effect as we think it should have. And it's probably because there's a hardwiring of biological systems initially that do change and can change, but in general, no. So we haven't seen it. Now, so I was just, so the paper from, or the opinion piece from the New York Times that was saying a little bit of alcohol is really detrimental, found about, they were citing a JAMA report from 2022 that said 142,000 people die a year because of alcohol consumption. Not necessarily because they're actually consuming the alcohol themselves, because, maybe someone else is consuming the alcohol, right, for different reasons, accidents, etc. So there's 100% some kind of bystander effect in that realm. But I'll do you one better. When I was a graduate student and learning about psychoneuroimmunology, talked to quite a bit of psychologists about bystander stress, caretaker stress, and both of those have an immense effect on your body, right? So if you're taking care of someone that's going through cancer, or if you're someone that just around someone else that's just high stress, it can be affected really dramatically. And there's two groups, one at University of Iowa, one in Emory, that really look into that. So hopefully I answered most of that.
I think that's a yes.
Yeah. Yeah, I guess I could have said that.
Great talk right here. I don't drink. It's just I'm nothing against alcohol. It's just my body doesn't have enough of the enzyme to break down alcohol. So with just 2 sips of just a beer, within 2 minutes you will know I've been drinking. So, but that saves me a lot of money over time.
Absolutely, headache too.
So my real question is, I look back at my grandfather's 80th birthday banquet. I was a kid running around, and I was surprised to see back then the servers were actually handing out cigarettes. And I was surprised looking back at those videos. So my question for you is, with the Surgeon General making that announcement, what's your prediction? How long will it be in the future days that we will walk into a wedding banquet where you have to ask for alcohol and not having just being served to everyone?
Okay, that's a good question. So actually, we were just talking about this in the realm the first thing was, how long is it going to take, in my mind, how long is it going to take for it actually to come into effect, right? Period, one. And then two, how long will it take for the public to really take, for cigarettes after that Surgeon General's note it took about 14 years, right? But now, I think really the biggest thing was the ban on people smoking inside that had the biggest effect of if you have to ask for a cigarette or if you're allowed to smoke or not. Not gonna lie, when I saw the Surgeon General do that with the notification, I looked at the price of a cigarette box, which is $9, and I was like, man, this handle of booze is gonna probably be like $90 from $30 in the next 5 or 6 years, and thinking to myself, maybe we should stop alcohol, right? Because I think that really the biggest difference is going to be people are just going to start making a lot of money. Because the folks that still drink will always drink, just like folks that smoke will always smoke. But yeah, that's a great social question that I'm guessing decade will come to effect, but it's hard, right? Because alcohol's steeped in history, So.
Yes, I have a question. I know you're in diet, you focus on alcohol. I'm just curious about this high fructose corn syrup. And it's possible to engage in the same kind of activity.
Yeah, that's a good question. So some of our diet studies have focused on the fats and the fatty acids that you find in some of the diets and the oils, but people do study both the fats and the sugars in diets, and it seems as if they do have an effect on changing pain sensitivity, but usually together, not necessarily by itself. Now, the high fructose corn syrup one, I can't tell you like with definitive, this is it, but for sure, like high sugars with high fats alter pain sensitivities.
What I was saying was a high fructose corn syrup, the gut microbiome and a lot of the immune cells are located in and around the intestinal system. But the change in the microbiome metabolism of this high sugar and the byproducts chemicals that it produces and its effect upon the central peripheral nervous system.
Absolutely. You said it perfectly, right? It's the fructose itself or the sugar itself, but then also the byproducts, right? Of how it gets broken down. And those actually are what we call inflammatory metabolites. So 100%.
Yes, sir. No alcohol involvement. Cancer patient taking immunotherapy. Is there such a thing as too much immunotherapy?
I was always in the camp of this could always be too much of anything, right? Depending on who you are, what you're exposed to, what you're dealing with. Right now, these checkpoint inhibitors in cancer immunology have been pretty big. They're cooling off, meaning more and more people are realizing that they are helpful, but they're not the completely cure-all, like end-all be-all to things. And so I think because of that, there probably is some sort of limit. But, for cancer, to me, always, what are your options, right? Like, do you want to just see what the fringe is going to get you, or do you want to just play it safe and not take the therapy, right? So, kind of get put in a hard place.
Well, the cancer treatment worked. Within six months, I was cured.
That's great. That's fantastic. Yeah. Congrats.
Online question, and this is really important. What are the difficulties that you need to overcome to apply this model with human clinical trials? What's the minimal sample size to generalize these outcomes? Because we're already starting to generalize it. How generalizable do you think it is?
Right. So I love that question. So in the lab, we always talk about the complexity of the human condition, and we often overlook how complex it really is, right? I had a couple questions about, what about this situation, what about that situation? And the defining parts of all that is the condition, right? Are you under a certain amount of stress? Are you eating a terrible diet? Do you already have genetic predispositions, right? And so for right now, what we can do is, do we see the same thing in the clinic, right? Can we generalize it, not overgeneralize it, but just generalize it? And we know that for a fact that there's differences in sexes between males and females, or females having more pain across chronic pain conditions, right? But that person's asking for some high level, like I need to break out like my experiential design calculator to get like a number, but like, I mean, for human, I think at least 1,500 people, to really start to get into the nitty-gritty of it all, with all the various factors that have to come into play.
How long away do you think something like that is to take this to clinical trials?
Most of this type of stuff is 15 years, right? So.
Are any clinical trials humans going on now?
Good question. So always, but not necessarily in this realm, right? And actually, I'm talking to a couple people. One of them, really good friend at NYU, who is a collaborator, and she's interested in female-specific differences in health disparities in fibromyalgia. But then when she collects patient data, and when she looks at her data, she gets people that are consuming alcohol at various levels and what have you. And so her and I were just talking about, is there a way to subset out her data to get some initial answers so that we can convince other people that it's worth the however many $5 million that we need to get started on it, you know, so.
We might have Dr. Philby, and you could partner too, someone closer. Just putting that seed in. Is there
I'm curious if you guys have looked at the age effects, particularly what would be the equivalent of like a teenage mouse in regards to depressive symptoms, but also pain intensity.
Good question. So these animals, the age equivalent would be about 21 to 28 years old, so clean adults. We had, I don't want to keep bringing back the grants. We had a grant that's in, that was supposed to get reviewed this week, that did get reviewed this week, that was looking at the effects of age. And so hopefully we'll get back in and talk about it. But hopefully, yeah, and well, we can, well, I'll tell you, we can do it in mice. So in mice, an aged individual or someone that's 65 or older is equivalent to about a 2-year-old mouse. And to keep a mouse alive for 2 years costs about $1,100 per mouse. So, like, it's expensive.
Quick question for you. How long does it take for that allogenic flow to return back to baseline?
Good question. I didn't talk about that at the time. Yeah, so it lasts for about a day, about 24 hours. I didn't mention it, and I thought, I was like, it's fine. But question, great question. It lasts about a day. And so, which is roughly a week in people time. So.
Have you done any dose ranging studies, Michael? Different doses?
Yes. Yeah, we've done, actually, that's another good question. Yeah, so we don't want to get too much higher than 5% because what we find is that at 10%, the animals just start experiencing pain-like behaviors because of the alcohol itself is just so much. But we've gone down to like half of what I've showed you today and then a quarter of it, and you see like a dose response of like less.
So it does affect those?
Yeah.
Curve.
Yeah, dose response.
Hey, I have a microphone. I'm thinking of Dr. Howard Sugar, John Sarno, Dr. John Sarno, the Curable app that is working now around pain. I've seen chronic fatigue patients, long COVID patients, fibromyalgia, POTS, join Curable to practice brain training. Seen a lot of recovery, especially in 20 and 30 somethings going on around pain. How do you think in polyvagal system trauma, comes into this fight, flight, freeze? How do you think that differs from what you're touching on here? And of course, many of these people have been off of alcohol, especially if they've been bedridden for years. But many are finding recovery this route. Where do you think that differs with this gene, you know?
I love that question. So I think
Yeah.
for us, right, the hope is that before you get to that severe point where the pain is manageable, that there is this inflammatory process that's happening and that checks and balances, that load is contained. And so we hope or we think that what we're finding is exactly the predisposition to these particular states that you find in POTS and fibromyalgia, et cetera. We have to, do more, think about it deeper, but we think that it's like the predecessor to all of it. And you bring up another point that I think is also important too, right, is that we would love to figure it out before it gets too far along the line, but there's always going to be a point where it gets missed, so you're going to need things like the app and other therapeutics as well.
Two more questions. Christy, is there one over there?
Oh, yes. So he was talking about the corn syrup, but I want to go back to the alcohol, because I've always heard alcohol turns into sugar. So was there insulin recorded in the male mice versus the female mice? And insulin resistance is all over, I've been reading everything about insulin resistance. So was that taken into consideration of the pain level, because the inflammation nodes were all over the brain of the female? So is that considered?
That's a great question. We did not look at insulin levels. We absolutely can, because we save all the different, what you need to measure it, but we didn't test it. And I think one of the reasons why we didn't think to test it is because with these short-term models, people don't see changes in insulin resistance. And what they do to test this is called a glucose tolerance test. Is similar to what we do with patients, right? Is inject the animals or a patient with glucose and test how long it takes for the glucose to go back down to normal. And I've seen that in a model similar to ours, but just much longer. And then we didn't test, we didn't look at it because we didn't think that it was going to change, but it's worth a try. So that's a great question. Yeah.
Thanks, Michael. I really appreciate that because, as you know, in human studies, we have to rely on subjective responses, and we can't often completely be 100% that we're getting sex differences based on just female being more introspective and so on than males. My question is regarding blood alcohol content, and thank you for showing those graphs there. I didn't catch whether the blood alcohol content was during the time of allodynia, or was it the titration to the 14-day kind of spring break model?
Gotcha. Good question. It was at all those, that's true. It was the blood alcohol was at the end of the experiment, so like right in the middle of when they're the most allogenic. So, but some of those measures, like the weights, were across the whole experiment, which were no different. And all the other measures are right in the middle of when they're experiencing the most pain.
Could it be just a metabolism effect then in females versus males?
Probably, 100%. I definitely think so, right? I don't, it's equal amounts of the ethanol in the blood, but how it gets metabolized can very well be different. And I think what goes to show that is the fact that when we look at I think it goes to the nuance of it all, when we look at the wild type animals, the males do have a baby response, right? But nowhere near as much as the females do. And when you look at when that gene is in those particular cells, that response completely goes away. And so there's something to do with not just where that gene is, but how whatever metabolites are making its way to that particular gene. So,
thank you.
Dr. Burton,
I get the last question, just because you talked about pain plasticity. We talk about neuroplasticity. There's some evidence that the female brain, especially during pregnancy, has a lot more neuroplasticity than any other time in our life. Do you think there could be something about there being more pain plasticity in the female brain?
Ooh, I like that. So I actually like to call our lab the Neuroimmune Plasticity Group.
Okay.
And I think it's interaction between not just the nervous system, but the immune system and the plasticity that's there. The reason why I think so is because immunologists have already shown, human immunologists have already shown that there's a discrete difference between the male and female immune system already, right, at baseline. And so that can have a direct effect on neural plasticity.
Thank you so much. I mean, don't you want to just sit at his table? We could spend all afternoon with you, I think. Because pain is, I mean, millions of people have it, and many of the people in this room are women. Thank you for such enlightening research and really changing the conversation about how we think about it.
Appreciate it. Thank you all.
We're so grateful to be here. So, join us next week. You won't want to miss Dr. Florence Chang. Her research is going to be talking about advanced brain imaging to find markers of neurodegenerative disease, as well as brain markers to treatment effectiveness. I also hope that you will sign up for our BrainHealth Presents, which is starting soon. It's registration. One of the speakers, while we remember, everyone's curious about their memory. One of the leading experts, Dr. Sharon Rangathan, is coming, and his book will be on sale. Everyone wants to know about their memory. And then we want you to sign up. We've got a new text challenge for brain health in terms of sleep. So do this QR code, pull out your phones right now. I want you can either type this number, 888-844-8991, and you will get the new text challenge. It will do it. It will go to the sleep challenge.
So the same number, but they text sleep?
Yes. Oh, they text sleep instead of gain.
Right.
Yeah, don't text gain, text sleep. But it's the same number. And every day you will get a new cue on how to sleep better. So we should look at the difference in sleep and pain and the immune system. Thank you, Dr. Burton. We will see you next week. Thank you.